At sexual maturity, each of a woman's ovaries contains about 200,000 immature eggs called primary oocytes. A primary oocyte is diploid and is arrested in prophase 1 of meiosis. A layer of follicle cells surrounds each primary oocyte. Together, an oocyte and its follicle cells make up a follicle. An ovarian cycle lasts about 28 days, beginning at the first day of menstruation or menses. During the first 7 days of the cycle, 6 to 12 primary oocytes begin to mature. As the follicles develop, the follicle cells communicate with oocytes and pass them nutrients through pores called gap junctions. Each oocyte grows larger and the surrounding follicle cells divide, proliferating to produce thousands of follicle cells in a single follicle. By day 7, all but one of the developing follicles begins to degenerate. The remaining follicle continues to develop, and its follicle cells continue to pump it with nourishment, and also supply it with proteins and informational molecules needed for early stages of development. The maturing primary oocyte completes meiosis 1 and divides into 2 haploid cells. Each of these cells receives half the chromosomes. However, one cell, called a polar body, receives very little cytoplasm. The other, now a secondary oocyte, enters meiosis 2 and arrests there until fertilization. At day 14, ovulation occurs, and the secondary oocyte erupts from the ovary. The oviduct contains microscopic cilia that beat and draw in the released oocytes. This immature egg enters an oviduct, where it may become fertilized by a sperm cell and complete meiosis. The follicle cells that are left behind develop into a small mass of endocrine tissue called the corpus luteum. The corpus luteum remains in the ovary for 2 weeks, secreting the hormones estrogen and progesterone. At the end of the ovarian cycle, if the woman is not pregnant, the corpus luteum disintegrates. Each month, the ovarian cycle is tightly coordinated with the uterine cycle. In the uterine cycle, the lining of the uterus builds up and then sloughs off. The cycle begins with the sloughing of the uterine lining. This is the first day of menses, also called menstruation. After menstruation, the uterine lining starts to grow again and to prepare for implantation of an embryo. During this phase of the uterine cycle, up until ovulation, the uterine lining proliferates. Capillary beds supply this tissue with nutrients. Just before menstruation occurs again, the capillary beds degenerate and no longer deliver the surrounding tissue with nutrients. During menstruation, this tissue dies and sloughs off through the vagina to the outside of the body. The ovarian cycle is controlled by the interplay of hormones from the pituitary gland and from the ovary itself. A few days before the beginning of the cycle, the anterior pituitary begins to increase its secretion of 2 hormones, follicle-stimulating hormone, FSH, and luteinizing hormone, LH. FSH and LH stimulate ovarian follicles to grow. As the follicles grow, they begin to secrete estrogen. During this phase of the cycle, the increasing levels of estrogen feed back on the pituitary to inhibit the release of additional FSH and LH. During the next week, the levels of FSH and LH drop. Beginning around day 12, the increasing levels of estrogen suddenly have the opposite effect on the pituitary gland. Instead of exerting a negative feedback on the pituitary, these hormones now exert a positive feedback, stimulating the pituitary to release FSH and large amounts of LH. LH reaches a peak at day 14 of the ovarian cycle. This LH surge triggers the mature follicle to rupture and release the egg, the process of ovulation. LH then triggers the remaining follicle cells to differentiate into the corpus luteum, which secretes estrogen and progesterone. The corpus luteum remains in the ovary, secreting estrogen and progesterone for the last 2 weeks of the cycle. At this point in the cycle, these hormones again inhibit the release of FSH and LH. A decline in FSH and LH restricts follicles from beginning to develop during the second half of the cycle. LH, or a hormone produced by an implanted embryo, is required to maintain the life of the corpus luteum. At the end of the cycle, if an embryo is not implanted, the corpus luteum degenerates. When the corpus luteum degenerates, it no longer releases estrogen and progesterone. The ovarian and uterine cycles are tightly coordinated. Hormones secreted by the ovary at different phases of the ovarian cycle trigger changes in the uterine lining. For example, at the beginning of the cycles, the levels of estrogen and progesterone are too low to maintain the uterine lining, and menses begins. About a week into the ovarian cycle, the developing follicle increases its secretion of estrogen, and estrogen levels in the body begin to rise. This hormone triggers the cells of the uterine lining to proliferate, and the lining becomes thicker. Just before ovulation, the level of estrogen in the body has reached its peak. Afterward, the follicle cells remaining in the ovary develop into the corpus luteum, a structure that releases estrogen and progesterone. The hormones maintain the uterine lining at a peak thickness and preparedness for embryo implantation. At the end of the cycle, if the egg is not fertilized or has not implanted, the corpus luteum breaks down and stops releasing estrogen and progesterone. Without these hormones, the uterine lining also breaks down, initiating menses.